Skeletal muscle tissue structure. So this is a structure of a skeletal muscle tissue, and so we can see that it is covered by an outer connective tissue layer. Here this is an outer connective tissue layer. It is known as epimysium, which gradually forms the tendon, and the tendon, it connects muscles to bones. Muscles are made up of fascicles. These are fascicles over here. This is one fascicle, this is another fascicle, this is another fascicle, and these fascicles are covered by perimysium. So you can look over here. This is the structure from outside to inside. The outer connective tissue is known as epimysium, and it gradually forms the tendon. Then we have muscles which are made up of fascicles, which are covered by perimysium. And many myofibers make up a fascicle. And myofibers are also known as muscle cells, and they have many nuclei and are covered by endomysium. So you can see here there's endomysium, it's the innermost. Myofibrils are bundles of myofilaments which consist of thick and thin filaments. Thick filament is also known as myosin, and thin filament is also known as actin. Now let us take a close look at the structures of actin and myosin. This is the outer to inner structure. Now this is the structure of actin, or the thin filaments. So the actin consists of two helically wound F actins. Now this is structure of actin, and they consist of, this is one F actin, this is another F actin. They are helically wound to each other, and these are made up of monomeric G actins. Each circle which make up an F actin is a monomeric G actin. And they also have troponin and tropomyosin. So these tropomyosin, they block the myosin binding sites on actin, and this troponin, this circular things you can see here, these are myosin. This is the binding of myosin to actin is blocked by tropomyosin, and tropomyosin are held together by troponin in place, and troponin has its own calcium binding site. Now let's take a look at the structure of myosin. So the myosin has a head. This is the myosin head. You can see here, here, here. This is the myosin head. It is a flexible hinge region and a tail. The myosin head is the most important part because it has actin binding site as well as an ATP binding site. So obviously in the ATP binding site, when ATP comes and binds and it gets split into ADP and inorganic phosphate, that's when the head rises up and goes and binds with actin. But actin cannot always bind with myosin because of troponin and tropomyosin. They are masking the sites where the myosin and actin can bind. And we missed an important structure in the previous slide, which is, you can see here in the thin filament, you can see something going in between the F actins. You can see one thin structure going in between the F actin. That is known as nebulin, and they connect the F actins together. Nebulin is associated with thin filaments and help to regulate actin assembly. So that's the function of nebulin. And this is how the actin filaments are in arranged along with myosin. The slide during the sliding filament theory and everything, when the muscles are moving, this is the part which moves. We'll see that in detail. Now we'll check the arrangement of filaments and proteins. So myosin, also known as thick filaments, attach on the M line. So this orange part, this is myosin. They attach on the M line. Whereas we have something else as well. See, we have actin, which is blue, and myosin, which is orange. So the actins attach to Z line and Z disc, and the myosin, the orange part, it attaches to M line. So the distance from one Z disc to another Z disc is known as a sarcomere, and they are the repeating units of myofilaments, and they shorten during contraction. The region containing only actin fiber is known as I band, and the region containing full myosin fiber including actin actin overlapped position is known as the A band. So here you can see that the Z disc is here, the M line is here, this is the myosin, this is the actin. What are these yellow structures? These yellow structures are none other than titin. They are elastic fibers, elastic filaments. They attach the myosin to the Z disc. So you don't want the Z disc to be sad that only actin is there. So even myosin are attached to Z disc via the nebulin, or sorry, the elastin or the titin filaments. Nebulin is found in actin, whereas elastin attaches the thick filaments to the Z line or the Z disc. So you can see here. Then there's something called the H zone or the H band. The region which is only having thick filaments, that is only myosin filament, is known as H band. And so here, when you take a close look, there's a region where there's both actin and myosin, and that is the zone of overlap. And there's what is the distance between Z disc to Z disc called? Yes, that is the sarcomere. So what intersects the sarcomere is the M band or the M line intersects the sarcomere. It is where the thick filaments, that is the myosin, attach. And the Z disc has attachment of both actin and myosin. It's not seen in this picture that well because the other thing which binds myosin to Z disc is nothing other than titin, which is an elastic protein. So you can see all the things clearly here. The H zone, where there is only thick filament or myosin. Z disc, where there's attachment of actin and myosin via titin. And there is I band, which is the light band, and the A band, which is the dark band. Now one technique to remember the I band and A band, that is the dark and light band, is A band and I band. A is dark band and I band is, forgive my handwriting, so A, I, I. So you know A band is dark band and I band is light band. I band is the band where only and only actin filaments can be seen, whereas A band is the band where actin and myosin can be seen, and that's why it's kind of dark. So thank you.